communication between the pre- and postsynaptic neuron is brief. It's important, therefore, to remove the neurotransmitter from the synapse. There are two main mechanisms for this to occur: reuptake and enzymatic deactivation. A reuptake transporter uses ATP to pump neurotransmitter from the synaptic cleft back into the presynaptic terminal. Enzymatic deactivation is where we have this Pac-Man enzyme that can bind to the neurotransmitter and facilitate a chemical reaction to break it down into parts called metabolites. This process does not require energy. There are two main reasons to remove neurotransmitter from the synapse. One, we want the communication to be relevant. If somebody rings your doorbell and your doorbell doesn't actually make a noise until 5 minutes later, that's not a meaningful communication. Or if somebody presses your doorbell and it continues to ring even after they've left, that is no longer a meaningful communication. It's only meaningful when they press the button, the doorbell immediately rings. That communication is meaningful. The other reason it's important to remove neurotransmitter from the synapse is that it can actually cause problems like excitotoxicity that can lead to death of the neuron. So when the neurotransmitter is released into the synapse, a number of things can happen. It can bind to postsynaptic receptors, fulfilling its role of communicating to the postsynaptic neuron. It can be eliminated through reuptake and/or enzymatic destruction, or it could bind to a presynaptic autoreceptor. These receptors are located on the presynaptic neuron and respond to the neurotransmitter that neuron releases. This is a negative feedback mechanism. It's like the presynaptic cell is telling itself to stop talking. The way this process works is that the neurotransmitter, in this case norepinephrine, binds to its presynaptic autoreceptor, which initiates a signal transduction cascade and inhibits the voltage-gated calcium channel. That means the next time the presynaptic cell fires, less calcium will come in, and therefore we'll get less vesicle fusion. We mentioned already the existence of axodendritic and axosomatic synapses, but there are other types of synapses as well, including an axoaxonic synapse. Here are two examples of axodendritic synapse from the terminal button of the presynaptic cell onto the smooth dendrite, or synapse from the terminal button of the presynaptic cell onto a dendritic spine. Axosomatic, as we said before, is where the presynaptic terminal makes contact with the soma of the postsynaptic cell. In the case of axoaxonic synapses, there are actually three neurons involved. Cell 1 is telling the terminal button of cell 2 not to talk to cell 3. Here's a closer figure for what an axoaxonic synapse looks like. So here we have terminal button A. This cell is the presynaptic cell. It's going to release neurotransmitter that will bind to terminal button B. It will cause a signal transduction cascade to decrease calcium entry through voltage-gated channels, decreasing neurotransmitter release from terminal button B onto the dendritic spine of cell C. Cell A telling cell B not to talk to cell C.